Hello students, and welcome to a special lecture on enzymes and enzyme catalysis. We are doing this particular exercise in this particular lecture because you might get asked some questions about enzyme kinetics on the final exam, or more importantly, your lab instructor may ask you to do some enzyme kinetics or do some plotting of enzyme graphs on the lab exam in week 12 of the course. So I'm just going to run through enzyme kinetics and the ability of enzymes to do their job in a little bit more detail. So first of all, enzymes, of course, are members of a class of chemical molecules called catalysts. Catalysts are molecules that speed up the rate at which chemical reactions occur, usually by lowering the amount of activation energy that is necessary in order to start the reaction. And the words catalysis and catalyze refer to doing this. They refer to reactions that are sped up with the help of catalysts. So here we have, any of you that have ever studied chemistry, you will recognize this as a reaction plot. This is where you have, you put together two molecules. An enzyme or a catalyst will start the reaction, and then you'll end up with some products which are different molecules than you started with. And you notice here that I'm just going to activate one of these pens. One of these highlighter pens. Notice that if you have no catalyst, you have to put quite a lot of activation energy in to start the reaction. If you have a catalyst, like an enzyme for instance, the amount of energy that needs to be put into the reaction to get it started is less. But in the end, the delta G, the amount of energy that is released by the reaction, is the same. But you just the reaction thermodynamics of the reaction didn't really change, except for the fact that it was easier to get it started in the first place. Now, enzymes, of course, are biological catalysts. So biological catalysts, they are catalysts that are made of protein, and proteins are encoded by genes. Of course, genes only encode proteins. Proteins are the only macromolecule that are encoded by genes. Okay, and we have quite a lot in the human genome. Many thousands of enzymes are encoded in the human genome. And when you name things in science, if we have a formal way of naming things, we call that a nomenclature. Right? A nomenclature is a formal way of naming things. Now, for enzymes, there are two ways you can name enzymes. One of them is a little bit of an informal way of naming them, which is you take whatever the substrate is. That is, the substrate is the molecule or molecules that the enzyme works on. So, for example, if the enzyme works on cellulose or amylose, then the substrate for those enzymes would be cellulose or amylose. Cellulose, if an enzyme that hydrolyzes cellulose, breaks apart the molecule by adding water to it. If the enzyme hydrolyzes cellulose, you simply take the name of the substrate in this case being cellulose, and you add the term the suffix ase to the end. So an enzyme that hydrolyzes cellulose would be a cellulase. An enzyme that hydrolyzes a protein would be a protease, and so forth. That's the informal way of naming enzymes. There is a formal way as well, and that is that all of the biochemists in the world get together every year, and they have a conference, usually in some place like Switzerland or someplace like that, and they decide to give every enzyme in the world, every known enzyme, a number, a coded number that means something very specific. And we're not going to teach you about that in this course because that's beyond the level of this course. But if you're interested in enzyme nomenclature and enzyme kinetics and the way that enzymes can vary reaction rates, then you should, if you're interested in that, you should take Biochemistry 201, which we teach here at the college as well. But anyway, that is the formal way of doing it, is to assign every enzyme a number. So the informal way, a cellulose enzyme an enzyme that hydrolyzes cellulose would be called a cellulase. An enzyme that hydrolyzes amylose would be called an amylase. Enzymes that hydrolyze lipids are called lipases. A phosphatase is an enzyme that removes phosphate groups from proteins, and a phosphate kinase is an enzyme that adds phosphate groups to proteins. You might have mentioned, you might remember that I mentioned earlier in the course that some proteins are turned on and turned off by either adding phosphate groups to them or by removing phosphate groups from them. So again, that falls under the purview of biochemistry. It also falls under the purview of cell biology. So if you're interested in that kind of stuff, you should take cell biology. That's Biology 200 here at Columbia, or Biochemistry 201, and you'll learn that there are all kinds of fascinating proteins that they are not continuously on, they're turned on when you add a phosphate group to them, or turned off when you remove a phosphate. So a very interesting study of how many proteins are regulated or controlled. Regulation usually means turning things on and off at the right time and the right place. Many fascinating proteins are capable of doing that. Okay, so here's the informal way of naming enzymes. Just add A's to whatever the substrate is that they work on. Here is the more formal way of naming enzymes. So a cellulase, instead of just calling it a cellulase, the International Enzyme Convention has given it the name EC 3.2.1.4. EC, and specifically it is called an exoglucanase. And I've forgotten my enzyme nomenclature. It's been a few years since I've had to use it, but I believe that like the numbers have a specific meaning as well. So I believe the number 3 means that whatever enzyme this is, it works by hydrolyzing its substrate, and then I think the number 2 means that it's an exoglucanase rather than an endoglucanase. An exoglucanase meaning that it's an enzyme that breaks down cellulose from the end, and therefore it's from one end and works its way inwards, and that's called an exoglucanase, as opposed to a cellulase that can start breaking down a cellulose molecule from an interior position, from the middle. We call that an endoglucanase. But anyway, we don't really care about that. Now down here in the bottom, you see that there's an interesting little thing that looks like a bunch of tubes and wires and ribbons. That is actually called a ribbon diagram, which gives you a rough idea of the shape of the enzyme. And shape happens to be very important to the activity and function of enzymes. Okay, so the properties of every enzyme, what it does for a living, are determined, one, by which amino acids make up the protein and in which order they are put together. So you remember, of course, that there are 20 amino acids. The 20 amino acids all have different chemical properties. So the chemical properties of an enzyme will depend on which amino acids you put together, and also in which order you put them together. But not only that, more importantly, the shape of an enzyme is important too. So as a protein, as an enzyme, or any protein for that matter, but in particular an enzyme, as an enzyme is being translated, which means that it's being converted from DNA into, you make DNA, you make an RNA copy of DNA, and then you take that RNA copy out into the cytoplasm of the cell, where it gets translated by ribosomes, an organelle called a ribosome, which sticks in one amino acid for every codon, every 3 base pairs. Okay, so once you put together the sequence of amino acids, they will also form a shape. They will, the different types of amino acids, many of them attract each other and many of them repel each other. And so those attractions and repulsions, most of which are hydrogen bonds, but there are a few covalent bonds as well, they also attract each other within the molecule, giving it another shape. Okay, so the shape of an enzyme is critical to its function, and you should know that if an enzyme accidentally loses its shape, we refer to that as denaturation. This is usually something that doesn't happen on purpose. So if an enzyme gets heated up too much because it's been left in a hot place or something like that, it will usually denature. If it happens to be, if it happens to be a sensitive enzyme that's part of the human biology system, that could be deadly, right? So that's why that's why humans and other animals tend to die in environments that are too hot, and the reason for that is because if the heat goes up too much, their enzymes denature, and then you can't generate energy, or you can't do other chemical reactions that are necessary. So denaturization is bad for enzymes. Obviously, usually you heat them up or you do something else that destroys their shape and their structure, and therefore destroys their function. Now, on the other hand, there are some times when you want to sort of change the shape of an enzyme a little bit. You change the shape of an enzyme in order to turn it on, to get it to start working, and then you change it back a little bit, change the shape a little bit to turn it off again. In that case, the change in shape, a deliberate change in shape that is critical to the function of the enzyme, is referred to as an allosteric interaction. So you should know what the word allosteric means. Allosteric refers to a deliberate change in shape of an enzyme or a protein in order to help it do what it's supposed to do. So there are many things that there are many enzymes and proteins whose activities and functions are controlled allosterically. Now, within an enzyme, we've known this for a long time in science, that a typical enzyme may be made of several hundred different amino acids, but only 2 or 3 of them are actually important. The other ones are just there sort of to fill space. And the 3 or 4 the 3 or 4 amino acids that are important to the functioning of the enzyme can be found in something called the active site of the enzyme, the active site. So an active site is the part of the enzyme that actually does the work. The rest of it is just along for the ride. It's just along for amusement. So enzymes typically have one active site where the reactions take place. The reactions that the enzyme is controlling or mediated, they take place in the active site. And an enzyme may have, specific enzymes have different numbers of active sites. So if you have one enzyme that has a single active site, it tends to work very slowly because it only has room to fit one molecule of substrate in there at a time. There are other enzymes that have 2 or 3 or 4 active sites, and they can do their work a lot faster because it's kind of like having 5 pairs of hands instead of 1 pair of hands, right? And in fact, in genetic engineering today, people who are doing biotechnology are deliberately engineering enzymes. You take an enzyme that sometimes you have, in order to do something interesting with an enzyme, you need 2 different enzymes, and each of those 2 different enzymes work separately, and each of them only has 1 active site. And with genetic engineering, these days, with genetic engineering, we can easily engineer our own tailor-made designer proteins and designer enzymes, where you have those 2 enzymes together on the same molecule, and then you've stuck in 3 or 4 active sites for each one of those independent reactions, and these enzymes can do their job much better than they could naturally. So we have the ability to do all kinds of fascinating things with making tailor-made or designer enzymes to do specific things. And that's something about biotechnology that doesn't get a lot of attention, but it is a very important endeavor. Okay, so now on naturally occurring enzymes, the place where the active site usually contains at least 3 amino acids, usually ones that are charged, and they're either acidic or basic, or you have 2 acidic ones and 1 basic one, or vice versa. And those three amino acids within the active site of an enzyme are sometimes referred to as a catalytic triad. Right, so if you ever hear the word catalytic triad, the term catalytic triad, that is what it's referring to. The idea that within the active site of an enzyme, there are usually 3 amino acids that are more important than all of the rest. As I said, for most enzymes, the active site is the only important part of the enzyme. And within the active site, you really have 3 amino acids that are critical to the functioning of that enzyme. The rest, the other 200 or 300 amino acids that make up that enzyme, are just there to fill space. All right, so let's talk about the structure that determines the ability of enzymes, or for that matter, determines the function or the utility of every protein. Okay, so number one, of course, is what amino acids is the protein or the enzyme made of, and in what order are they put together, and what shape do they assume once they're assembled? Again, that's an example of allosterics. All right, proteins or enzymes, which are a type of protein, they have several levels of shape that form the structure of the enzyme and that we have to know. All right, so the primary structure of a protein or an enzyme is simply the amino acid sequence. So what amino acids did you put together in what order? We refer to that as the primary structure of an enzyme. Now, once you put together a chain of amino acids, it's a fact that the amino acids all have different chemical properties. Some of them are positively charged at neutral pH, some of them are negatively charged at neutral pH, some of them are acidic, some of them are basic, and so on and so forth. And so some of them tend to attract each other, and others tend to repel each other. And when that happens, the result of that is that the enzyme or any protein for that matter will assume certain smaller structures at the local level. So certain parts of the protein will assume the shape of something called an alpha helix, and some parts of the enzyme will assume a structure called a beta sheet or a beta pleated sheet. So those are the two most common secondary structures among enzymes, alpha helices or beta sheets, and I will show you what those look like in a minute. Okay, next we have the tertiary structure. Tertiary means the third level of structures, and that is, okay, so you put together this amino acid sequence, and then certain parts of it formed into a helix and certain parts of it formed into sheets. So you have several helices and several sheets together. What organization, what structure do they assume altogether? Right? And that's called the tertiary structure. And then finally, we have the quaternary structure. Quaternary is the fourth level of structural organization. So generally, in biology, you have primary, secondary, tertiary, and quaternary refers to 4 levels of organization for things, not just necessarily proteins, but in this case we happen to be talking about proteins. So the quaternary structure in this case it refers to enzymes that have more than one subunit. They have more than one peptide chain. Remember that a peptide is a short protein, so you should know that the word peptide is the word that we use to describe a small or a short protein. So generally, if a protein is maybe up to 100 amino acids or something like that, we don't dignify that by calling it a protein, even though it is, but we don't dignify it by calling it a protein, we call it a peptide. But to be a real protein, to be a real professional protein, you have to be 300, 400, 500 amino acids in size. So anyway, if you have a protein or a peptide that has only one amino acid chain, then we only have to worry about primary, secondary, and tertiary structure. However, there are many, many enzymes where you take 2 different amino acid chains and put them together after they've assumed their primary, secondary, and tertiary shapes. You put them together. And so there are some enzymes that only have one subunit. There's some that have 2 or 3 or 4, and so that refers to the quaternary structure. Is there more than one subunit to the enzyme? And finally, there's something we have to worry about called cofactors or prosthetic groups. You might remember in class that I mentioned in class that if you take a man and you cut off his leg, like he loses his leg because of an accident or something like that, and they put on an artificial leg, what do they call that? They call that a prosthetic limb. So the word prosthetic, literally, the word prosthetic means that it's a piece of the whole, but it's not made of the same material. So if you put a wooden leg onto a man, the man is made of proteins and amino acids and whatnot, but the leg is made of wood. It's made out of cellulose, so it's a prosthetic. It's an integral part of the whole, but it's not made of the same material that the rest of the unit is made of. So that's what a prosthetic group is, also known as a cofactor. Now, in enzymology, the cofactors are often metal ions. So you know for a fact, or you know, for instance, that hemoglobin is a protein that carries oxygen around, and hemoglobin has 4 subunits. So hemoglobin has a quaternary structure, and more importantly, every one of those subunits has an iron ion, coordinated iron ion in the center of the molecule. So hemoglobin could not carry oxygen without the iron prosthetic. Right, so the iron is considered to be a prosthetic group. The iron is located inside a chemical group called a heme group. But the best example, or the easiest example to remember of a prosthetic group, is just remembering the fact that hemoglobin is an enzyme that carries oxygen around the body. It could not do so if it didn't have an iron group in the middle of a prosthetic group called a heme group inside. Okay, so now here's two terms that you should be familiar with: apoenzyme and holoenzyme. You need to know these two words. The word apoenzyme refers to an enzyme that does not have its prosthetic groups, right? So that doesn't happen in nature very often, but it happens a lot in pharmaceuticals. So like if pharmaceuticals, biochemists use enzymes all the time, and sometimes if you look in the freezer of a laboratory in the refrigerator, you'll see there's a bottle that lists the name of some enzyme, and then it calls it the apoenzyme. And then you know that if you see that, you know that if you mix that enzyme up in some water and you put it into a chemical reaction, it won't work, it won't do anything, because it needs a prosthetic group first. So you have to mix it with some magnesium, or you have to mix it with some iron or something that's essential for the prosthetic group to do its job. And when an enzyme has all of its prosthetic groups, if it has the iron that it needs, or it has the heme molecule that it needs, it is referred to as the holoenzyme. Okay, so starting with primary structure, we put together the amino acids one by one, as shown here, and we make a chain of amino acids, right? So reading from left to right, the writing is very small here. I apologize for that, but it looks to me like starting from what's known as the amino acid end, which is seen at the top of this chart, it appears to say glucose or glycine, proline, threonine, glycine, so on, right? So the names of the individual amino acids are listed in all of those little blue circles. So this is the primary structure of the enzyme. Okay, now once that primary structure has been formed, some of the amino acids will be attracted to each other and some will be repelled by the other ones, and that will cause them to spontaneously form something called an alpha helix. On the other hand, some of the other amino acids will end up attracting and repelling each other and forming a sheet called a beta-pleated sheet, which is a different type of a structure. Okay, so the alpha helix and the beta-pleated sheet are the two most common examples of secondary structure of a protein. And an individual protein will have usually will have a dozen or so alpha helices spaced throughout the protein and a dozen or so beta sheets spaced throughout the protein. And so in order to see how those things all interact with each other, where are all among the general shape of this enzyme, where are the alpha helices located and where are the beta sheets located? So in order to look at that, we have to ask what is the tertiary structure? What is the tertiary structure? And in order to visualize that, we draw something called what's called a ribbon diagram, right? So this is a ribbon diagram of an enzyme, and these sort of coil shapes here, these red coil shapes, are the alpha helices that are formed when that protein is translated. And these green arrows, these kind of thick ribbony arrows, are meant to be sheets of beta material, so beta pleated sheets. So this is a ribbon diagram where we have several helices, the red things, and we have several beta pleated sheets, the green things, and then these other lines that connect them are considered to be disorganized. They have no consistent shape. They're just kind of holding the helices and the sheets together like they were pieces of string or something like that. But they are flexible. They're flexible. Okay, so this is a ribbon diagram like this is an example of a tertiary diagram, or it's an example of the tertiary structure of a protein or an enzyme. Here's another one that we just looked at. This is the exoglucanase that I showed you earlier, and suddenly this picture makes more sense, doesn't it? So you can see all of those red cylinders on the left. Those are a whole bunch of alpha helices, and then you see some blue ribbons on the right. Those are a bunch of beta pleated sheets, and then the lines, the squiggly lines joining them, are areas of disorganization where the molecule has no consistent shape. All right, so we talked about quaternary structure and prosthetic groups. So here is hemoglobin, and notice that hemoglobin is made up of 4 individual globin units. Right, there's 4 of them. And then within each one of these subunits, you have a coordinated heme group. That's what these kind of reddish brown things are. And then within each heme group, you have an iron ion, right? So hemoglobin happens to be an example of an enzyme that has a quaternary structure. It has 4 subunits, and each one of those subunits has a prosthetic group inside it, a heme group, which is a chemical compound, and then at the center of the heme group you have an iron ion. Okay, and here we have an example of an apoenzyme versus a holoenzyme, right? So you see on the right, there's a diagram of hemoglobin without the heme group in it. So it's kind of hard to see on these colors, but let me see if I can. No, that doesn't work. I was trying to draw a white line around the cofactor down here. This cofactor, that is a heme group with an iron ion. If you take that out of the hemoglobin molecule, it can't carry oxygen. So you might see a bottle in the refrigerator in the lab that says hemoglobin apoenzyme, which means that if you want to use it to carry oxygen, you have to add some heme and some iron to it. So remember the words apoenzyme and holoenzyme. Apoenzyme is an enzyme without its cofactors. Holoenzyme is an enzyme with its cofactors. Okay, now before we go any further, I should mention to you what something called a chelation factor is. Okay, so the word chelation refers to grabbing something. It literally translates from Latin as to grab something. And a chelation factor is a chemical that we use in the laboratory, and there are several of them, so I'm not mentioning any particular one, but there are several of them, and what they are is chemicals that have the ability to basically grab and steal the prosthetic groups out of an enzyme. And when you do that, you stop the enzyme from functioning. Right? And so there are many times when you're doing an experiment and with an enzyme and you want to suddenly stop it from working at a certain time, and so you would add a chelation factor. Now in the biology lab, when you did the lab on amylase enzymes, you took 6 test tubes that contained amylose and amylase enzyme, and you let the amylase enzyme hydrolyze the amylose for a few minutes, and then you stopped the reaction by adding iodine. So like you had 6 tubes and you let the reaction continue for 1 minute, and then you stopped the reaction in one of the tubes and set it aside, and then you waited for 2 minutes, and then you stopped the reaction in the second tube and then put it aside. You waited 3 minutes and you stopped the reaction and put that tube aside, and then 4 or 5, and then you could measure the amount of starch or amylose that had been digested per minute by looking at those 6 test tubes individually. And the way you stopped the reaction was you added iodine to it. Right? So iodine is a chelation factor for the amylose enzymes. It stops the reaction from occurring. In the lab, there's a more common one called EDTA. That's an abbreviation for a chemical group, but EDTA is the most common chelation factor that we use in the laboratory to stop a reaction. But in the biology lab earlier in the semester, you were stopping the amylase enzyme from digesting the amylose by adding iodine to it. Now, one of the reasons why we like to do that is because iodine will not only stop the reaction, but it will also react with the amylose and change color. Any chemical that changes color is called a chromogen, by the way. I wouldn't ask you that, but on a test or anything, I wouldn't ask you that. But iodine happens to be a chromogen that binds to the amylose and turns dark bluish brown. Right, so the amount of the amount of dark bluish brown color that you have in the test tube is proportional to the amount of amylose starch that you have there, right? So when you stopped one of the tubes after 1 minute and you put the iodine in, it turned dark brown because most of the amylose was still there. Then you had the next tube that you stopped after 2 minutes. It was a lighter shade of brown because during the extra minute, the enzyme had time to digest or to hydrolyze more of the amylose. And then after 3 minutes, you had an even lighter shade of brown because the amylase enzyme had digested or hydrolyzed more of the amylose. So that is the explanation of how the amylose experiment that you did worked. You timed it by taking 6 tubes. You stopped one of the reactions after 1 minute, one of them after 2 minutes, 3, 4, 5, and then you, with the iodine that you added, you looked at the color and you used that color change to estimate how much of the amylose had been digested in that time. So that was the point of that. All right, now enzymes can be slowed down by things that either alter their structure or do other things, but allosteric interactions that slow down or inhibit enzyme function are very common. All right, this is just demonstrating this is what's known as a ball model that looks like it's made out of little balls. This is demonstrating that here we have an enzyme, and there is the active site right there, and there's the substrate, whatever it is this enzyme works on. And so in order for the reaction to take place, this substrate has to end up somehow in the active site, and once it's in there, it can be digested or hydrolyzed. This is another diagram demonstrating putting together two substrate molecules in the active site of an enzyme, and then once the enzyme catalyzes this reaction, it releases the products, and then the enzyme is recycled again so it can be used again. All right, now what sorts of things can inhibit an enzymatic reaction? Okay, so I'll mention competitive inhibition and non-competitive inhibition. So remember that inhibition means to slow something down or to stop it from doing whatever it wants to do. Inhibits, when you inhibit something. Okay, so competitive inhibition is where you put your enzyme in with the substrate, but there's something else in there that will also fit into the active site, but it's not a substrate, right? So when you put that into the substrate, you're kind of wasting your time because you put a molecule into the substrate that can't be turned into the products you want, right? So that is called competitive inhibition because the substrate that the enzyme is supposed to work on, the substrate and the inhibitor molecule are both competing for a space in the active site. Right? So if the inhibitor gets in there, the enzyme can't do anything with that. It has to kick it out and wait for a proper substrate molecule to come in. So that's the difference between competitive and non-competitive inhibition. There's another thing called that was competitive inhibition that I just explained, right? So when you have something that fits into the active site but it's not a substrate, that is competitive inhibition because the actual substrate and the inhibition molecule are competing for a spot on the active site. Non-competitive inhibition is where there is a molecule that if it binds to the enzyme, it will inhibit it from doing its job, but it does not bind in the active site. It does not bind in the active site. Instead, it usually binds someplace else. It attaches to someplace else on the enzyme and causes an allosteric change in shape, and that allosteric change in shape inhibits the activity or the functioning of the enzyme, right? So that would be an example of non-competitive inhibition. Thirdly, there's something called feedback inhibition, and that is where an enzyme produces a substrate, or it creates a molecule, and that molecule, the product that was created by the reaction itself, will inhibit the enzyme, right? So for example, there's a bunch of there's a bunch of enzymes that make the molecule, the amino acid tryptophan, and those enzymes that make tryptophan can be actually inhibited by tryptophan itself. So the enzymes will make a bunch of tryptophan, and then when the concentration of tryptophan in solution gets high enough, the tryptophan will actually turn off the enzymes that create it. So it's, for now we don't need any more tryptophan. When the tryptophan that we've created gets consumed, then those enzymes will turn back on again, because the tryptophan that was holding on to the enzyme will come off in the lower concentration. All right, finally, we need to be aware of a concept called reversible and irreversible inhibition. Reversible inhibition is where you have an inhibitor molecule that either binds to the active site, competitive inhibition, or binds somewhere else on the molecule, non-competitive inhibition. And so in reversible inhibition, that inhibitor molecule binds to the enzyme, stops it from doing its job, but it can come off again. Under the right circumstances, it will come off again. And then in irreversible inhibition, you have a particularly nasty inhibitor that either binds to the active site or binds somewhere else on the molecule and changes the shape, and it never comes off. It never comes off. So essentially it has deactivated, permanently turned off the enzyme. Okay, so a good example of reversible inhibition is the example I just talked about with the enzymes that produce tryptophan. So the tryptophan molecule is created by an enzyme, and then when the tryptophan molecule is created, the tryptophan molecule itself binds to the enzyme and turns it off. But when the supply of tryptophan in the body drops to below a certain level, the tryptophan that's inhibiting the enzyme will come off again, and it will make more of the product. Okay, irreversible inhibition, a lot of the poisons that kill people or animals or whatever are irreversible inhibitors of enzyme function. So for example, you may know that oxygen is carried by hemoglobin, and carbon monoxide is a gas that's produced by internal combustion engines like car engines or tractor engines and so forth. And you know that if you inhale, you probably know that if you inhale too much carbon monoxide, it will kill you. The reason for that is because carbon monoxide competes with oxygen on the hemoglobin molecule. The difference, of course, is that oxygen is reversibly bound to the hemoglobin molecule. So oxygen, when the hemoglobin is in your lungs, it picks up an oxygen and then it circulates through the blood, and then it delivers that oxygen to another part of the body. So the hemoglobin, the oxygen will stick to the hemoglobin when it's in the lungs, but it will come off again when it delivers the oxygen to wherever it's supposed to deliver it to. On the other hand, if carbon monoxide binds to hemoglobin, it will never come off. Right? So you've heard of people dying every year from carbon monoxide poisoning. And if somebody gets carbon monoxide poisoning, you can't fix it. You can't cure them of carbon monoxide poisoning. Let's say you see somebody who's suffering from carbon monoxide poisoning. You can't really heal them by giving them oxygen, because the carbon monoxide has bound to the hemoglobin and it won't come off. So the only way to treat them is to give them a blood transfusion. So you give them fresh blood that does not have carbon monoxide attached to the hemoglobin molecules. So that's an example of irreversible inhibition of enzymes. All right, so here we're just demonstrating. Here we have an enzyme that has an active site, and the active site is right in the middle there. And it's called the capsase 1 enzyme, by the way. And then there is an inhibitor. The substrate molecule binds to the active site, but so can an inhibitor molecule. So if that inhibitor molecule gets into the active site, it will block it, and the entire enzyme will be inactive. Now, usually that's a reversible inhibition, so the inhibitor molecule can come back out again, and the enzyme will work again. All right, so here we have an example of non-competitive inhibition. So here's the active site of this enzyme. Here's the substrate that is supposed to fit into the active site. right? And here we have a competitive inhibitor which binds to the active site and prevents the substrate from binding there. But this particular enzyme also has the ability to be controlled using non-competitive inhibition. So here we have a non-competitive inhibitor that binds to a different spot on the enzyme, but that binding of this molecule here changes the shape of the active site so that the actual substrate cannot bind to it anymore. So that's it. This, what I'm showing you here, is actually an example of an enzyme that can be controlled through both competitive inhibition and non-competitive inhibition. Here's another example of an enzyme that is just regulated by, controlled by non-competitive inhibition as well. So there's an allosteric, what's called an allosteric binding site. An allosteric inhibitor binds to that site, causing a change in the shape of the active site, which prevents the actual substrate from being able to bind to it anymore. Here's another example of the green thing is meant to represent an activator. So here we have an enzyme that is actually inactive unless an activator enzyme binds to another spot on the molecule, binds to an allosteric interaction site, and activates it. And then there is also an inhibitor that can bind to a nearby site and change things for the worse so that it's inhibited. Here's an example of a series of enzymes that produces the amino acid threonine, and it operates through feedback inhibition, right? So if you don't have enough threonine in your body, these enzymes will work to make some threonine. So here's a substrate that is, I can't remember which molecule it starts out with, but it's able to convert that molecule into threonine. So we have the substrate which gets converted into threonine, or sorry, we're converting, I'm sorry, I see what we're doing. We're converting, changing threonine into isoleucine, which is another amino acid, right? So this enzyme causes threonine to be converted into isoleucine. Isoleucine then comes and binds to the enzyme that created it in the first place and stops it. Right, so if we have enough isoleucine, we don't need any more. It stands to reason that the isoleucine will circle back and inhibit the enzyme that created it because it's not needed anymore. Right, so on the other hand, well, anyway, that is an example of competitive inhibition. All right, now enzymes do their best work under a set of optimum conditions, and so that is what we're going to discuss next. So every enzyme works at an optimum. Optimum means the best conditions, the best, right? So every enzyme operates best at an optimum temperature, an optimum pH, and an optimum substrate concentration. The temperature is easy to understand. Usually if the temperature is too low, the molecules that are in the reaction, they don't collide with each other as quickly, and so the reaction slows down at a temperature that's too low. That's fairly common. At a temperature that's too high, the enzyme starts to denature. A higher temperature causes an allosteric change in shape, which can lead to denaturation. Right, so you should be aware that temperature, that is how temperature can vary the activity or the speed at which an enzyme does its job. If the temperature is too low, naturally all reactions occur at a slower rate if the temperature is low. But if the temperature is too high, the enzyme will start to denature. The same thing is true for pH. For pH, the charge on the amino acids that make up every protein will vary depending on the pH. So if you change the pH, that too will alter the shape of the enzyme, and it will cause the enzyme to denature a little bit. It will cause an allosteric change in the shape of the enzyme. All right, now the substrate concentration, this is a little bit interesting. What's happening is that if you put an enzyme into a solution with some substrate and the concentration of the substrate is very low, the enzyme has to sit around for a long time, bored, twiddling its thumbs, waiting for a random collision with a substrate molecule. And when a substrate molecule randomly collides with the enzyme can do its job, and then it has to sit around waiting until the next substrate molecule comes along. Okay, so you can see how if the concentration of substrate is too low, the enzyme cannot do its best work. The enzyme cannot work as fast as it otherwise would. Then if you add more substrate to the solution, it can work faster. But then you can imagine that you get to a point where it can't work any faster even if you give it more substrate. So at that point where the enzyme has just enough substrate so that it's working at its maximum speed, at its maximum capacity, that is considered to be the optimum substrate concentration. If you increase the substrate concentration beyond that, the enzyme can't work any faster because it is saturated, right? And so that would be a kind of a waste, wouldn't it? That would be a waste. So that's actually what the optimum substrate concentration will be, that concentration where the enzyme is working as fast as it can, and then adding more substrate will not increase the rate at which the enzyme is doing its job. Okay, so how do we determine this, right? So we do some experiments where we hold the temperature and the pH constant, and we vary the amount of substrate, and then we do another experiment where we vary the temperature, but we hold the pH and the substrate concentration the same, and basically we hold two of the variables constant while we vary the third variable. We manipulate the third variable, and that's how we figure out what the optimum constant or what the optimum conditions are for a particular enzyme. So here somebody's done an experiment where they vary the temperature, and while varying the temperature, they measure the reaction rates of the enzymes, and then they can use this to determine what temperature gives you the optimum or the fastest rate of reaction. Right? And so here we have the optimum temperature for the human, a typical human enzyme is 37 degrees, which should not be any surprise to you because that's human body temperature. So enzymes in the human body do their best work at 37 degrees because that is the body temperature, human body temperature. If you go into a very hot climate where the ambient temperature is 45 or 45 degrees, which it happens more often these days because the planet is heating up, if you go into a very hot climate, you can't take that for very long because the enzymes will not work as well. So you can see here that the activity for human enzymes drops off rapidly at about 40 degrees. And so 45 degrees, 50 degrees, you can't live very long in that temperature because the enzymes in your body begin to denature at that temperature. But, lower than 37, they slow down, but not dramatically. Above 37, the enzymes will start to denature. Now, this other line, the red one, is from a bacteria that is called a thermophile, a thermophilic bacteria that has adopted, it is adopted to live at a temperature of 77 Celsius. And so the optimum temperature for those creatures, for the enzymes inside these thermophilic bacteria, is around 77. Okay, what about pH? Okay, so here is somebody did a similar experiment where you vary the pH and then you measure the rate of the reaction. Right? And then here we have the optimum condition, the optimum pH for enzymes in the stomach is 2, and that's because the pH in the stomach happens to be 2. It's a very acidic environment. On the other hand, trypsin is a protein that operates in the intestine. The intestine is an alkaline environment, which is very basic. And so its optimum temperature is 8. And so the human stomach has a pH of 2. The human intestine has a pH of 8. The enzymes that you find in the stomach versus the intestine are tuned accordingly to work best at those pHs. And here is our friend, the substrate concentration. So you notice that if you put an enzyme, in this case, the enzyme is called lactose hydrolysis, lactose hydrolase, and its job is to break down the carbohydrate lactose. And so you notice that if you give it you vary the amount of substrate that it has to work with, and then you measure the reaction rate here. And so At low substrate concentrations, the enzyme is not working as fast as it can because it has to wait for a random collision with a substrate molecule. But as you increase the amount of substrate in solution, the reaction gets faster, faster, faster. And then about here, when this line starts to flatten out, the reaction will not increase by giving it more substrate. And the reason for that is because it can't work any faster. It's already working as fast as it can. So that area right there, say 30 millimolar of lactose, is the optimum substrate concentration. Here's an interesting graph where they try to, this is a three-dimensional graph where they show temperature, pH, and substrate concentration all on the same graph, and then you get a kind of a three-dimensional surface. So on a three-dimensional graph like this, you just want to look for the highest point. So I would say the highest point is somewhere in there. All right, now let's do an assignment today. We're going to do an assignment, and let's determine the optimum conditions for an amylase enzyme. Okay, and also, why don't we, while we're at it, let's compare the optimum conditions. Let's figure out the optimum conditions for an animal amylase, like a human amylase, and compare those to amylases from plants, because plants typically have a different body pH. They have a different ambient temperature than humans do, for instance, and so we wouldn't expect an amylase enzyme from a person to have the same kinetics, the same enzyme kinetics or characteristics as an amylase from a plant. But let's find out. All right, so under what conditions do enzymes do their best work? We know about this, right? So they have an optimum temperature. If the temperature is too low, the reaction is slowed down. If it's too high, the enzyme starts to unfold, which is also called denaturation. The same thing happens if you vary the pH. If the pH is too high or too low, the enzyme will start to denature as well, and it will have an optimum substrate concentration. Let's learn how to draw a graph, except that in science, of course, we don't say that we draw a graph, we say that we plot a graph, right? So let's learn how to plot a graph. First, a variable is something that you vary, of course. Right, so variables like time, temperature, reaction rate those are examples of variables. But we define one of the variables as the dependent variable, and the other one we define as the independent variable when we're comparing two variables. So for instance, if we're trying to figure out how fast an enzyme works at a different pH, we're going to say that the speed at which the enzyme works depends on the pH. Right? Also, if we want to determine the optimum temperature that an enzyme works, we're going to say that the reaction rate for that enzyme depends on the temperature, doesn't it? Now, it's not the other way around. The time the time doesn't slow down or anything depending on the reaction rate. It's the other way around. So the reaction rate will depend on the amount of time that's passed, or the reaction rate will depend on the pH or on the substrate concentration. So one of the two variables we're looking at is dependent on the other. So you should know the difference between, you should be able to tell me which is a dependent variable and which is an independent variable. You also know that the graphs that you've been seeing, they consist of a vertical line and a horizontal line. Right? Now, by definition, the horizontal line is called the x-axis, and the vertical line is called the y-axis. And by definition, the independent variable always goes on the x-axis, and the dependent variable always goes on the y-axis. Right? So if we were going to plot enzyme activity versus pH, we would plot the pH, we would put the pH on the x-axis, and we would put the reaction rate on the y-axis. So what I've done here is I've done that, and what we're going to do here is let's start out with a simple example. We're going to measure how much, we're going to put an amylase enzyme in with some starch, into a starch solution. And the starch solution, the enzyme will hydrolyze the starch as the reaction proceeds, right? So we're trying to figure out how much starch has been hydrolyzed over time. So we've put time on the x-axis. Time is the independent variable because the amount of starch that is hydrolyzed depends on how long the enzyme has been in there with the starch, doesn't it? Right, so then we'll put the concentration of amylase in solution on the y-axis like so, and we see that at 0 minutes we have a 10 millimolar solution. At 1 minute we have a 9 millimolar solution, and after 2 minutes we have an 8 millimolar solution, and so forth. And from the slope of this line we can calculate the reaction rate. Now, just for simplicity, because we're doing this experiment to look at how fast the enzyme works over time, we've held the temperature constant at 20 degrees Celsius and we've held the pH constant at 7. We're going to analyze the optimum temperature and the optimum pH later, but right now we're just looking to see how fast the enzyme works sort of generally. So we determine the enzyme rate by dividing the rise in the line by the run. All right, so after 5 minutes, 10 millimolar of the amylose has been hydrolyzed. So 5 millimolar in 5 minutes, that comes out to 1 millimolar per minute. 1 millimole per minute is the rate of this enzyme at 20 degrees Celsius and pH 7. Okay, now instead of plotting how much of the amylose is left per minute, we could also have plotted this graph so that the line is rising, right? And by saying how much was digested, right? So we could basically flip this graph the other way so that the line is going up instead of down by asking or by plotting how much of the amylose has been eliminated or digested or hydrolyzed per minute as opposed to how much is left per minute. And so that's really what you did. That's more or less what you did in the lab. You did an experiment but usually that's what we would do. We would measure how much of the enzyme has been digested per minute. What is the digestion rate per minute? All right, so different enzymes from different sources have different sets of optimum conditions. We explained that already. So we're going to calculate how fast under what conditions does these two enzymes, a plant enzyme and a human enzyme, do their best work. Okay, so here we have a plant enzyme, a plant amylase, and an animal amylase. So can you tell me these questions down here? What is the dependent variable and which goes on the x-axis? And by the way, we're going to do this part of the experiment at 20 degrees in pH 7 and a substrate concentration of 10 millimolar. Okay, so which is the dependent variable? The dependent variable is the amount of amylose remaining, right? If we're just going to do the graph like we did before. And the independent variable is the time, of course. The time is going to proceed regardless of what the enzyme is doing. The march of time is not dependent on how fast the enzyme is working. How fast the enzyme is working or how much of the amylose has been digested or destroyed or hydrolyzed is dependent on the time. Okay, so the time will go on the x-axis. The amount of amylase remaining will go on the y-axis. Okay, so now I'm going to look at at time 0 here. I see that the plant amylase is at 10. Okay, so there's the plant amylase. And then at 10 minutes, at 10 minutes, which is here, the amount of plant amylase remaining is about 9. At 20 minutes, it is 8, right? Something like this, right? And then at 30 minutes, it's at 7, so it would be there somewhere. So we've plotted the points. We can now connect the points with a line, right? So that's basically what the plant amylase looks like. Now let me change colors here. And at 10 minutes, at 0 minutes, the amount of plant enzyme, sorry, the animal enzyme at 0 minutes is 10, as you would expect. And then after 10 minutes, we're down to 8. Right, so I'll put another point there. After 20 minutes, we're down to 6. At 30 minutes, we're down to 4. And I won't plot all the points in this case, but as you can see, the amount of amylose left in solution drops more quickly in the presence of the animal amylase, which indicates that the animal amylase is working faster than the plant one is, right? So we've distinguished between the two enzymes using 2 colors, but we could have just used 2 different symbols. So I could have, plotted X's for the plant amylase and circles for the animal amylases. So you might have to do that on a lab exam or something like that, where you draw 2 different lines. You need some kind of a legend that tells you which line is which, and you can tell which line is which by using different symbols when you plot it as well. Okay, let's try it again. This time we'll measure how much, we'll instead of measuring how much hasn't been hydrolyzed, we'll measure how much has been hydrolyzed. Okay, so there's our data. All right, so let's take a look at the the plant amylase again. So at 0 minutes, 0 millimoles of the amylose has been digested, has been hydrolyzed. At 10 minutes, 1 millimolar. At 20 minutes, 2 millimolar. At 30 minutes, 3 millimolar. Right? And you can draw all the rest of the lines in if you want. I'm just going to show you that connecting them gives you something like that. And then I'll change colors again, and we'll look at the animal amylase, right? So it's 0, 0. This is called the origin, by the way, where it's at 0, 0. And then at 10 minutes, 2 millimoles of the amylose has been digested. At 20 minutes, it's 4. At 30 minutes, it's 6. Right? So I won't draw all of these, but it looks, again, like the animal amylase is digesting the amylose faster, and so you get a steeper curve, and you can, from the slopes of those curves, as I showed you before, you can measure them again, the rates again. So this time, instead of measuring how much amylose is still remaining that hasn't been hydrolyzed, we're measuring how much has been hydrolyzed. And this is the reaction rate for the amylase, or the animal and the plant amylase. Okay, what about the optimum temperature? Right, so let's have a look at the plant amylose. Right, so we have temperature down here. And then instead of amylose concentration, we're giving the reaction rate over here. Okay, so at 0, 0, the origin. Then at 10 minutes, the plant amylase, has reduced, has digested 3 millimolar. Right, so there, right, and then 20 minutes, 10. Sorry, wait a minute. 20 minutes. It has actually hydrolyzed 10 millimolar of the 10 millimolar, right? And then at 30 minutes, we're down to 4. And at 40 minutes, we're down to 3. Right? So what this is showing me is that if you connect these dots as best you can, that's showing me that the plant amylase has an optimum pH, an optimum temperature, has an optimum temperature of about 20 degrees. Now let's look at the animal amylase. So 0, 0, we'd expect. Then at 10 degrees, we have 2. Right, so 10 degrees, we have 2, rate of 2. At 20 degrees, we have a rate of 5. Where are we here? 5. At 30 degrees, we have a rate of 10. And then at 40 degrees, it drops all the way back down to 1. Right, so the animal amylase has a peak, has an optimum temperature of around 30. if this graph had had better resolution, if we'd plotted more points, you would have seen that it had an optimum temperature of 37. Right, so that is how you draw that graph. All right, then you do the same thing for pH. In fact, we barely really need to plot the graph. You know what it's going to look like. It's going to tell us that the optimum pH for plant amylase is around 10, and the optimum pH for human amylase is around 8. Not 10, 5. Right, and the optimum pH for human amylase is about 8, which is exactly what you'd expect. So you can plot that onto the graph. I won't take up any more time by doing it, but you get the idea. And then substrate concentration, right? So without even plotting the graph, you can see that the curve kind of flattens out here, right? So probably the optimum substrate concentration for plant amylase is about 6 millimolar or 7 in there somewhere. And the optimum for human, you notice that the curve starts to flatten out here. So the optimum for humans is probably about 5. So that would tell you so your graph would look something like this, right? So it would look something like that. With the plant amylase leveling out at 5, and then the human amylase leveling it out at 6, something like that. So I'll let you draw that one in. And once we determine those, from those 3 graphs, we determined that for the plant amylase, it has an optimum temperature of 20, an optimum pH of 5, and an optimum substrate concentration of 8. For the animal amylase, it has an optimum temperature of around 30. In fact, we know that it's probably 37. Optimum pH around 10, so it's being digested in the intestine rather than the stomach. And it has an optimum substrate concentration of 6 millimolar. All right, so what we're going to do with our assignments is I'm going to give you an assignment, a homework assignment to take home and do where you plot 3 graphs for temperature, pH, and substrate concentration to determine the optimum pH for an enzyme. You bring that back to me. If you get everything correct in that exercise, you get 2 points. And next week, when you bring it back to me, you give me the one back that you did for homework, and we'll do one live in class. And if you get that one correct, you get 3 more points, and then I will add, of those 5 potential points that you could get from these 2 exercises, I will add them to your midterm score, because the computer has, the Moodle computer has nowhere for me to put a bonus mark. So if I give you a bonus mark for some assignment, I'll add it to the midterm mark. So I'm going to take whatever mark you get for these 2 assignments and add that to your midterm mark, and that is where it will be. Okay, so good luck with that assignment, and good luck with the rest of the course.